Good morning. This is Penelope Perkins Veazey. I'm a professor at North Carolina State University, and today I'm going to be presenting the section on the post-harvest working group that goes with the Growing New Roots Grafting SCRI grant. This will cover the last 4 years of this particular grant. The objective of this particular aspect was to look at post-harvest quality of resulting grafts for the Solanaceae crops and Cucurbitaceae. In this case, Solanaceae choices ended up being eggplant, pepper, and tomato, and these experiments were done in Washington State, Kansas, Florida, Ohio, and North Carolina. Cucurbitaceae in this particular part of the grant ended up being on watermelon only, and this was also done at Washington, Florida, and North Carolina. Going over the results from Washington State University, here verticillium wilt is a major problem with establishment of eggplant and pepper. So 14 rootstocks for eggplant and 3 for pepper were obtained from the World Vegetable Center and used to look for resistance of verticillium wilt. And the collection of fruit from these scions, which were millionaire for eggplant and ace for bell pepper, were also checked to see that there were any negative attributes from the grafting. And in this case, there were no differences. There are similar fruit size and qualities such as dry matter and firmness and pericarp thickness for both the eggplant and the pepper. Moving on to Ohio State, here the peppers were looking at pungent pepper versus bell pepper. In some earlier work, pungent peppers were found to have some really excellent disease resistance, so they were very useful as rootstocks. But the next question mark was, would this transmit pungency to bell pepper? So the two components, the capsaicin and capsiate are in, capsaicin is the pungent part that gives pungency to the hot peppers, and capsaicin is found in bell pepper. They both have anti-inflammatory effects. However, pungency can be a really negative turnoff for people who don't like hot peppers. So the idea was to look at the quality of these peppers and also secondary metabolites. These are the types of pungent peppers that were used as rootstock on the left, and on the right, Aristotle and Carolina Wonder were used as a bell pepper. So in the first part, which was using the pungent pepper as a rootstock, there was a slight increase in soluble solids and decreased acidity when grafting was used. But there was no increase in capsaicin of the scion fruit. So the pungency from the rootstocks was not being transmitted in any sizable quantity into the bell pepper, at least not enough to pick up when they were looked at for sensory attributes. And in fact, there were also no differences in color. Now, the opposite is also underway, where the pungent pepper is being used as a scion and bell pepper is being used as a rootstock to see if you get any attributes in that fashion. Moving on to the tomato work, in North Carolina State, a large field trial was done for 2 years to look at the effects of pinching, which was to remove this main stem and control, utilizing some vigorous and less vigorous rootstocks, and also incorporating a less vigorous scion, which is Tasty Lee, and a vigorous one, which is Mountain Fresh. And the yield and marketable yield are talked about in another part of the project, but here the tomato organoleptic properties are discussed. So the grafting made no differences as far as the pinch and control part for organoleptic. There was no difference in the fruit quality. Overall, the differences are slight in terms of organoleptic. The Tasty Lee seemed to be a little bit more responsive to grafting than Mountain Fresh, and the acidity and soluble solids were a little bit lower in Tasty Lee, but the lycopene was higher when it was grafted to Shield, which is a less vigorous rootstock compared to non-graft. So what does this mean? Not really clear. It's a little fuzzy as to why 2 aspects of ripeness would be a little bit less, and yet lycopene, which also indicates ripening, would be a little bit higher. At Kansas State, Tasty Lee was also used. In this case, it was grown under a high tunnel system. And the idea was to also look at organoleptics and look at something to do with the carotenoid profile, not just lycopene, but other components of this. And what happened was that when Tasty Lee was grafted, the fruits were bigger. When it was grafted, especially to Maxifort, the fruit size was increased a bit. If you look at the overall components, though, organoleptic, it was a little less clear. In the non-grafted fruit, And a couple of the rootstocks tended to have lower titratable acidity than the other fruit from the other rootstocks. The total soluble solids is a wash. It's between 4.4 and 4.9. It's possible you could pick up a slight taste there because anywhere you need about a 0.5% difference in Brix for people to possibly start picking up a taste difference. Lycopene and beta carotene. Lycopene was slightly lower for non-grafted in a couple of the rootstocks compared to the other rootstocks, and beta carotene tended to mimic that. Beta carotene is oftentimes a little bit lower when lycopene goes up because the two interact. Florida had 2 large trials that were done on tomato, and the first trial was specifically look at quality of the fruit post-harvest. So in this case, there was an organic production system that was done both in the high tunnel and in the field, using beefsteak and grape tomato as their scions, and a number of rootstocks, the vigor versus less vigorous type. And here, these were looked at in terms of a number of attributes, all the quality aspects such as color, firmness, fruit composition, also a storage study, and also consumer sensory analysis. The upshot of it was that there were no differences in most of these except for soluble solids being slightly lower by 0.2% in the garden gem, which is the grape tomato, compared to the non-grafted fruit. Storage study, the quality time, post-harvest time was the same. There were no differences in sensory analysis. Consumers had the same acceptance, juiciness, etc., flavor. This was done also with the garden gem compared to the graft, and the non-graft were similar. In the second big study that was done at Florida, this was also a high tunnel production into organic systems using grape tomatoes, determinate and indeterminate types to look at, and again on these types of rootstock to see what would happen now, fleshing this out a little bit more. And in this case, 2 transplanting dates were incorporated, also an early and a late. So what happened was that the late transplanting date generally had a negative effect on the fruit, possibly because of more disease load by the time the fruit were mature. So the fruit in this later transplanting date tended to be less sweet, and the consumers were able to pick up that they were less sweet. There were some interesting interactions that happened. The rootstocks that were used in the early transplanting date all reduced fruit firmness in these grape tomatoes, but did not have the same effect on late transplanting. Possibly the grafting fruit matured slightly faster compared to the non-grafted in the early transplant date. We don't really know for sure. Now moving into the cucurbitaceae, in this case, this is all watermelon. This is all seedless watermelon of different scions were used across the U.S. In the first study that was done at Washington State, there were 3 studies that were done. The first study, all of these used Secretariat as a scion. The first one was looking at germplasm accessions from USDA, and Tetsuka Buto, which is an interspecific squash hybrid. In this case, no differences were seen in soluble solids, pH, lycopene content, rind firmness, and thickness among the grafted ones. The non-grafted plants died from verticillium wilt, so there was no way to tell a difference on these. The second study was using black and clear plastic mulch, and in this case, it incorporated the maturity factor that where you can leave a watermelon in the field 7 to 14 days after it's considered at full maturity to see what happens to the fruit. In this case, the rootstocks were Lagenaria, which is bottle gourd, and also one called wax gourd, Benicassa, and a couple, again, of interspecific hybrids. Super Shintosa has been used quite a bit over the years. The quality aspects for watermelon were followed, which included things like hollow heart ratings, hard seed count, solids, and total soluble solids and lycopene content. There was no difference with the mulch type, the rootstock treatment, or the harvest date, but the flesh firmness was found to be higher for watermelon grafted under Super Shintosa compared to non-grafted watermelon. So we have that interspecific hybrid effect again, but only with Super Shintosa. In the third trial with Secretariat, Tetsuka Buto was used again, and also Shintosa Camoforce, another interspecific squash rootstock. And 2 drafting methods were looked at. Again, these were left in the field for up to 2 weeks after fruit maturity. And here, the harvest date was the same for all of them, but those that were left in the field for 14 days were heavier. So they gained a little bit more weight by leaving them there. The attributes of hollow heart and hard seed and total solids were the same for all the treatment, but they did say that there was a slight increase in using the Shintosa camelforce only with a splice grafting system. Fruit were firmer than non-grafted watermelon. And lycopene was highest at 7 days after maturity in the field. Florida did work using Melody seedless, and in this case the idea was to look at the interspecific hybrid, squash hybrid, and also a straight squash, C. machata rootstock. Many times the complaint has been that using a squash as a rootstock could cause off flavors in watermelon. So attributes of rind thickness, firmness, flesh color, etc. were also looked at, and additionally consumer sensory tests were added, looking at the flavor, overall appearance, and acceptability, and the just about right scale was included. And in this case, no differences were seen. It was not negatively impacted, even with just a straight C. machata rootstock. But there was the trend again to see increased flesh firmness when using a squash or interspecific squash hybrid rootstock. Lycopene content was in and out depending on which treatment was used. The good news is that consumers did not show any major reduction of fruit quality when using these squash rootstocks. And then the final study done with watermelon with North Carolina State was to look specifically at what was going on at the cellular level, particularly with cell walls. And there are two aspects to this. One was to look at hollow heart formation. Can grafting reduce hollow heart? Which seems to be, there seemed to be a little bit of data to indicate this could happen. And also when we use this interspecific rootstock, squash rootstock, can we try and nail down a little bit more about what might be going on at the cell wall level? So using hollow heart, this is a pretty big field trial. It was done twice. And what happened was that when you use carnivore interspecific rootstock, you reduce the hollow heart by 34%. We were able to induce very nice numbers of fruit with hollow heart, but we saw a very strong attribute of reducing hollow heart by using the rootstock. Firmness of the flesh was also increased by about a half, this is in newtons, but this translates to about a half a pound of firmness. However, the other compositional parts of this that were looked at, the citrulline, the arginine, lycopene, soluble solids, pH, etc., did not differ between the grafted and not grafted fruit. The student who was assigned to this, a graduate student, their next job was to look at the cell wall aspects of grafted versus non-grafted, and also hollow heart versus no hollow heart. So using confocal microscopy, she had to look at the heart tissue from grafted and non-grafted with and without hollow heart to see if cell sizes and density changed. And basically, she was unable to show that cell density increased with the grafting. However, when looking at hollow heart, she did see that there is 20% of the hollow heart cells were bigger and also had less density, which would be expected if you have larger cells. And this supports some work that was done earlier with light microscopy. Looking at cell walls, there are several components of cell walls that can be looked at, such as cellulose, hemicellulose. Pectin was the one that was focused on here, mostly because this is the one that's most likely to degrade first in cells-to-cell stickiness, and is thought to be most likely to cause loss of firmness. When one looks at the fractions to see what components of NutriSugars are in each fraction, only the carbonate fraction was higher with grafted. It was 27% versus 25%. The pectins, the neutral that were present, they can be there as freely bound. So they bind wherever they want to on the pectin backbone. Also, there are major sugars such as homogalacturonin and rhamnogalacturonin 2. These are high components of the pectin backbone, interspersed in the backbone, and then also the backbone itself, which is made up of a number of neutral sugars bound together. Really, the only thing we found that was different with grafting for this was one neutral sugar. Of the 34 neutral sugars that we looked at, only allose was higher, and it was twice as high in grafted as in non-grafted. Now, this was a bit of a interesting conundrum because it's like, okay, we're seeing an increased firmness phenotype and flesh firmness, but we're not necessarily seeing a direct line to anything at cell wall level, at least in terms of pectin, although we did see generally an increase in neutral sugars. But the allose part was interesting. There's not much known about allose. The only thing that we were able to find was on rice. And it evidently can act to gibberellin as a hormone, and it can inhibit the gibberellin action on cells. And gibberellin tends to expand or elongate a cell. And in the case of rice, this is very important for disease control. It may be that in watermelon, by having more allose, it's slowing down the watermelon cell expansion, and this could help both with hollow heart formation and also tissue firmness. So a summary of all of these trials, what we saw was that when we looked at overall changes in organoleptic properties and sensory tests and even carotenoids, we found very little difference with grafted versus non-grafted fruit. The impacts seem to be more clear on survival in the presence of disease pressure and less about fruit quality. And this probably speaks to the good match of compatible rootstock and scion. You get your crop, and your crop is acceptable yield, and your crop is acceptable quality. In the watermelon trials, these are all using seedless scions of different rootstocks, different places across the country, but generally we saw a trend that the interspecific rootstocks do seem to increase flesh firmness in seedless watermelons. But the increases in the other attributes, such as the composition of the fruit, are not consistent, and they depend a little bit about where the work is done and about what was done. The good news is the sensory attributes appear to be unaffected by grafting, so we're not going to get, hopefully you're not going to get off flavor using the types of interspecific rootstocks that are right now that are available. There were a number of graduate students trained in this grant cycle, including 3 from Florida, 2 from North Carolina, 2 from Washington State, 1 from Ohio State, and 1 from Kansas State. So what are new areas of exploration that could be looked at? We still are having problems with off flavor in specialty melons such as Gallia. We don't know why. It could be a grafting compatibility, a simple grafting compatibility such as phloem xylem, or it might be more involved, such as at the RNA side, where the messengers that are, or transcription of important enzymes and substrates is not going the way it should be going to avoid these off flavors. It would be interesting to know if we could use genomics to look for possible post-harvest outcomes, to use it as predictive tool. And it would also be interesting to look at the junction of scion and rootstock to see if we can track down cellular molecular changes, enzyme activities, certain hormones, et cetera, that are altered by messages either from the scion or from the rootstock. What other ideas are out there? What do you think that needs to be done? Have we matured on our post-harvest aspects, or is there any room for other things that could be done? Could we be going a little bit beyond not just production and making sure that we have good fruit quality, but could we start trying to reach a little bit longer or reach a little higher for longer post-harvest life, a possible post-harvest tolerance of abuses such as temperature or too low a temperature or too high a temperature, to see if we can get a more consistent quality over this long-term period. Particularly the fact that we can increase firmness in watermelon may be useful. And also, should we be utilizing genomics and bioinformatics? We have a watermelon genome, we have a tomato genome, we have a pepper genome. Can we start using these and dig a little bit more to look for metabolic events that appear to be related to grafting itself? That's all I have.